Hello, everybody. Good evening. Welcome to the webinar on accelerated drug screening using 3D modeling. I'm your host, Shekhar, and I wish to start today's webinar with the word disease. As biosciences superheroes, super scientists, you'll always find yourself gravitating around diseases. And that obviously means looking after, looking for drugs, looking for bioavailability of drugs, looking for pharmacokinetics of drugs, looking for stability of drugs, looking for the toxicity of drugs, and multiple aspects of drugs. And in fact, drugs are the super weapons we all have as biosciences heroes, super scientists in our quest, in our war against diseases. Hello and welcome to Biotechnica and this webinar powered by Altem. We are into the fifth session today. And as I come to you live from Bengaluru, capital city of Karnataka, I have only one thing in mind. How can we empower the young generation, young scientists, young superheroes of today and tomorrow? How can we equip them with more tools so that they are empowered to take on the battle, whether it is coronavirus or any other future pandemic causative pathogen? My dear friends, it gives me immense pleasure to invite our guest of honor today, the guest speaker, Devya Shanmugarajan. She is here once again to enlighten all of us. But before I pass on the controls to her, as always, I wish to request you, please go ahead, share this link on Instagram, WhatsApp, whatever social media you use, LinkedIn, whichever is your choice. Please share so that the world knows what we're talking about today, so that we all can come on the same page and develop a great platform called as Biotechnica. So today, as we had informed you via email also, we have a few mega announcements to make. The first is that upon several requests, the Altem team has agreed to conduct a special Q&A session separately. So that will be a separate Q&A session apart from the 6 sessions. And there, all of you, whoever have questions related to Bioprinting, so that will be a closed webinar, which will be announcing shortly. The next mega announcement for all of you is Biotechnica. So from our side, we have decided to select 5 active participants among all of you. Okay, 5 active participants who have attended all the 6 sessions, including today and tomorrow. Including today and the next session. And you will be receiving a customized Biotechnica t-shirt. Okay, so that's about a prize from our side. 5 active participants among all of you. So all you have to do is be more active, okay? Ask more questions, okay? And the third and very important announcement is there is a bio quiz series going on the LinkedIn page of Altem Technologies, okay? I am sure most of you have already participated. So Altem Technologies is giving away an amazing voucher of rupees 2,000, okay, to those who will answer all the 5 bio quizzes correctly. Okay, so go ahead and participate in the bio quizzes right away. Okay, the link will be given to you in the description shortly. Okay, so these are the things, these are the mega announcements which we have. And now we are ready to start our battle against diseases with today's webinar, right? So quickly in chat, if you guys could, let me know if you are eager to learn. If that is the case, then type ETL in the comment box. Okay, ETL. Let's see how many of you are eager to learn. How many of you are eager to proceed forward? ETL, yes, wonderful. So this is what pumps us all up. That okay, now is the time to start. So, ladies and gentlemen, boys and girls, all the super scientists of today and tomorrow, I invite Devya S. for this session as our guest speaker. Devya, over to you. Divya, I guess you need to unmute your mic. Your mic is muted. The mic is muted. Yes. No, in the console. You have to go into the console and unmute it. Open the browser. Yeah.
Is it fine? Is my
Yes.
voice is audible? Perfect. Thank you so much.
Yes. Now you can bring back the PowerPoint.
Yes.
Yeah.
Perfect. Thank you. Is my screen is visible right now?
Yes, it is. Go
Yes,
ahead.
thank
You
you
can start.
very much. Yes. So, very good evening to everyone, students, scientists, professors, and ego research scholars for attending a wonderful session of Altem Technologies. I think this is the fifth session. We are sponsoring along with an Altem Technologies Biotechnicum, and very interesting topic. And you all know about that now the technology is moving from an 4G to 5G, and also there should be an improvement, and we should move forward from a 2D models to 3D models. So for whatever we are doing discoveries in vitro technology or more into 2D models. But that is actually, if you look at the previous sessions, I explained more about why we need a 3D bioprinted disease models to accelerate our discovery process. Now I'm going a little bit deep into it, or funneling down why we need to accelerate our drug discovery screening with the 3D models. So we are 3D peoples, right? And our system has been covered in such a way that it will try to interact with all the cells, and then communication will happen from one cell to another cell. So all are interconnected. And when we try to do it in a 2D model, only in a monolayer, that doesn't going to give us a perfect results. And more assays, and when the drug is getting into the market, most of the drug gets fails. Why it is happening? So though we followed in a perfect steps, and we followed a methodology, and we had a very good SOP to do that, still we have an failures. How do we prevent that failures? So this discovering a process with the 3D models will give you a perfect solution for you all, and give you insight knowledge about why we need your drug screening with 3D models. Let's continue with it and walk along with me and learn more about an accelerating drug discovery process with 3D models. And we are here from Altem Technologies. So we know about our company. If you have been attending our previous sessions, you come to know about our company very well. So Altem is one of the leading company in a 3D printing technology, and one of the things in your life sciences is the Dashall systems, where we've been introducing a BioVR Discovery Studio and Material Studio, and more into bio 3D printing with a cell link technology, where we introducing extrusion-based printers, light-based printers, and where it will be using an DLP technology and so on. And you can see our esteemed awards has been given for us. You can see it right hand side, sorry, left hand side. So as keep moving on, drug discovery. So getting directly into topic. We know that everyone will try to introduce a drug discovery, especially if you are a chemist or a medicinal chemistry or a pharmacy people or life sciences, whatever it may be. We will try to introduce or come to emerge with some kinds of drugs or lead molecules. As I told earlier, 90% of drugs entering the clinical trials will fail to meet the market. Why it is happening? So that's I told you, right? We are trying to introduce all the perfect steps we are following, and there is no wrong in whatever the methodology that we are following. Everything seems to be perfect. Why the drug is failure? Just keep thinking about it. I will give you the solutions at the end of the session. On average, you know about it, if one drug needs to be coming to enter the market, we have to spend nearly 10 to 15 years. And average cost of one drug to bring into market, right from the beginning till the end of that FDA approval, it needs nearly a $770 million need to be spent for one particular drug. So what if it gets failures in the intermediate state, or in a preclinical testing or post-clinical testing or in a phase clinical trials? So whatever the money we spend or energy we spend, time we spend, it is going to be a waste. So how to address this issue or how to overcome this issue? And is there is any way? Of course, there is a lot of way. As the technologies keep moving on, there is a solutions for all the problems. So if you just dive more into it, you can see the drug discovery and development. So first, what we try to do is we try to identify the disease. So any disease, whatever the disease is, maybe a corona, in a pandemic, if you think about it, we got the vaccine in a very shorter period of time. How it is happening? It's all introducing often a computation biology and chemistry and utilizing a 3D printing models. So first, what we need to know about it, we need to identify the disease. What is the disease? How it is happening? And then you have to know, isolate the protein. Which protein is majorly involving in causing the disease that we need to identify. So in general, it will take nearly 2 to 5 years. So how do we cut these years and enhance or accelerate our drug discovery process? There is a solution. Just wait and see as I keep working on slides. And once it finished, once you know which protein is causing a disease, what we try to do, we try to explore the proteins and try to understand the behavior of the proteins and networking, what are the network of proteins are involved into it, and how to resolve. And we also looking into a metabolic pathway, so and so lots of things and parameters that we need to keep eye on when the researchers are going to work on it. Then you will identify this is going to be druggable side, and we are going to design a drug, and then it will going for preclinical testing, and then going for an animal model in vitro testing, scaling up formulation, then only it will go for the FDA approval. So when you take this one, it almost takes a minimum 10 to 15 to 16 years to get a drug into the market. So where even though the drug comes into the market, lots of failures will happen. Maybe it is because of genetic patterns or maybe environmental conditions. So many parameters are actually involving in this process. But in spite of all those things, we need to achieve the efficiency of the drugs to be in a maximum. How it's possible? There is a way that it can be done through one of 3D printing technology utilizing 3D models. Instead of, since we are a 3D model, if you try to do this analysis in a 3D model, that will be a great solution for to accelerate the drug discovery process. So how to do that? Is there is any methodology? Is there is any protocol? What we need to follow? Let's walk along with me and know about it. So there is a convergence. Drug discovery is not a single domain that one person is going to, only one domain is going to involve. There are certainly lots of domain is going to involve. Say for instance, if you want to start from a beginning, we have to design. If you are an chemist, we know the organic synthetic route and we'll be synthesizing using a combinatorial chemistry, and then we'll be deriving many molecules. And we'll identify not all the molecules that we are discovering or being used in sketching, and we'll be testing, we're going to have the same activity that is called structural activity. So physiology, biochemistry parameters, and everything is going to play a major role. And when we come into properties that you want to test, how what is the behavior of your drug, we need to consider the whole physiology, pharmacology, immunology, and so on. And further moves on to the first kinetic studies, area properties, and so on. So this pharmacokinetic properties are very important that we test about, and or how much absorption is happening, distribution is happening, and how much excretion is happening. If drug is going to stay inside, and what is the excretion or toxicity property of the drug. So if all the parameters are need to be analyzed. And then you also need to check an in vivo activity, behavior, pathology, physiological behavior. So male, female, age group. So all those things all need to be considered when we test or when we have to do this in vivo activity. Then comes to safety, how much safety the drug is, and whether there is no side effects, and physiologically good, and safety assessment need to be done before going for FDA approval, and designing, and as I told, the combinatorial chemistry, still from a combinatorial chemistry, and also once a compound is going to be a novel and we are trying to propose a new biological activity, the intellectual property rights and patent rights need to be addressed. So this is not an individual domain, but it is combination of or convergence of our various disciplines to discover a one particular drug to be coming to the market. So, of course, as I keep on telling a convergence, there should be a convergence of all the disciplines. So we call as a bioconvergence. This bioconvergence will help you to accelerate the drug discovery process. If you accelerate the drug discovery process in a shorter period of time, then if you know what is the disease and the symptoms, the treating of the disease and then the pathology of the disease treating will be very easy. So why we need bioconvergence? Let's see this very small video. It is going to tell us in a very big details about why we need a bioconvergence to accelerate the drug discovery process.
Over the last century, the human race has made tremendous strides in healthcare, driving forward the quality and length of life on this planet we call home. Despite these extraordinary advancements and scientific breakthroughs, there are massive burdens on our healthcare systems, and we still find ourselves treating the symptoms rather than the disease.
So this is absolutely true. We are trying to treat the symptoms rather than a disease, but how to address these issues?
These challenges require a more holistic approach. Bioconvergence is where nature and science intersect, where advancements in biology and technology can unlock the door to things like personalized medicine and organ regeneration. As the leading bioconvergence company, Cellink offers exactly that. Technologies, systems, materials, and expertise in the key areas of the bioconvergence revolution equip researchers to answer the most pressing questions and enable this brighter future.
So it will help you to understand much more about what are the technologies and what steps need to be followed. And CellLink will provide you the tools for a bioconvergence workflow. Let's see this workflow.
Cellink provides the tools for a bioconvergence workflow from early stage development to large-scale manufacturing. Such tools provide the potential to help doctors customize cancer treatment through omics-based tumor profiling, miniaturized drug delivery, and tissue replacement. The potential to enable the regeneration of neurons and tissues for treatments that can change lives. The potential to measure the activity of a patient's muscles and brain as they go about their daily lives for early detection of neurological diseases such as Parkinson's. By democratizing advanced research tools, we provide researchers with a fresh perspective to find answers and empower them to leverage the potential of precision engineering, automation, and artificial intelligence. This will propel us forward to the future, a future where personalized medicine is no longer a dream, but a reality. Join us on this journey to create the future of medicine.
So personalized medicine, I think for past decades we have been keep on speaking about a personalized medicine because each people will have a different behavior to drugs, and then the symptoms and everything will keep changing from some group of people to other group of peoples, and then the personalized medicine, especially the people who is having an adverse effects of a disease who need to be treated immediately within a shorter period of a time, that comes into a show of a precision and personalized medicine. That is no longer dream when we try to introduce these technologies into a research process and to improve for a scientific research to achieve this. So as I stated earlier, this is the steps that we try to follow in a discovery process, discovering preclinical testing, phase 1 trials, phase 2 trials, and phase 3 trials, FDA approval, and post-marketing. So where we can reduce this speed or accelerate this discovery process. So first of all, when we want to test in a lab and animal testings, so this can be cut down or can be reduced by introducing a bioprinting within our 3D models that will help you to understand more reality about our in vivo system. So when we're testing our in vivo system very precisely in that particular phase, then the success of your drugs to be entered into a phase clinical trials phase 1 phase 2 will be more of certainty will happen, so that will enhance your discovery process. So instead of 16 years of spending a time, that can be reduced 2 to 3 folds, so the drug will be entered into the market without having any failures. So we're introducing a 3D models in drug discovery. So this is providing a new tool for everyone, and also it will prevent your late stage clinical failures. So why it is happening? We know that I already been keep on stating that we'll be using our 2D self-culturing technology to understand about the behavior and the molecular pathogenesis and the network of how the drugs are going to interact or going to given a behavior, or we are going to measure the bioefficacy on that. But however, preclinical testings with this going to be failure, but it can be improved or it can be enhanced by introducing in your 3D rather than a 2D models. Because as I stated, keep on stating that 3D based having external signals from your cells and the environment, which is more likely equivalent to your systems. So instead of before testing into animals, if you're going to utilize the 3D models, that will help you to accelerate your drug discovery process in your right path, and achievement of results is going to be 100%. So why 3D cell culture model? If you didn't attend our previous session, this is going to be a glimpse of this. So it supports to be a more cell-to-cell interactions. And you can see the morphology of the cells and cell proliferation and how the cell, whatever the metabolism is going to happen, it is all going to represent as like our in vivo system. And moreover, oxygenations, drug intake and nutrition uptake, and then interactions and sending a signaling, and everything else matters a lot when we introduce in a 3D cell culturing technology, and that can be done only through a bioprinted within a 3D cell cultures and extrusion and junction proteins. So all these things that when you're trying to give exactly like a in vivo systems, it will help a scientist and researchers to have a more physiologically relevant models. And also one can study the longer term influence of the disease and the side effect of the disease also. Because as we go for 2D cell culturing, at one period of time there will be depletion of nutrients and there will be having a toxic effect over the cells. But that can be overcome if you're going to use a 3D cell culturing model. And moreover, the disease progression and the screening of drugs for a longer time, that will help you to understand more about the drug behavior also. So absolutely, now we can know that instead of going for 2D, transit from a 2D to 3D cell culturing technology and to accelerate your drug discovery process. I think now you find the solutions why we need to go for 3D instead of 2D technology. So, generating, this is the first step. First, how do you do this process? It is most important that we are now. I think people or audience are so eager to know how do we do this process. So, answer is the bioprinters. The bioprinters will help you. Is exactly as I stated. It's a bioprinter. It will behave and give an environment as like our biological systems, and it is going to be automated, and it can have an because our system is very complex. We have a cells connected with the blood tissue, nerves, and everything. So all are being very complex network of tissues. So one can have an automated fabrications of any kind of a tissue textures and architectures that can be done by utilizing the bioprinters. Luminex, as is a BioX, will help you to generate the disease models. So how it will help you to generate your disease models? You can tailor for a specific organ types, and more physiologically relevant disease models can be done. And even if you want to do it with the healthy tissue model, yes, we can do that. So you will see that when we do any kind of 96 well assay, 100, 536 well plate assay, we will try to generate disease models and we test it how much efficiency of the drugs are there. But when we try to do this, we can come to know live behavior of the cells. At the end of my session, we'll come to know how lively this technology is capturing day by day, minute by minute, or second by second changes in the cells that are happening. And the high throughput screening can be done. I was being extremely saying about 1100 536 well plates. First we can start with the 96 well plates or 384 well plates, and you can use a dispensing technology and print it with the bio cells, and you can have this technology to generate disease models. What disease model that you want to generate, that will be keep cracking your mind. Any kind of a disease models that we want to explore and we want to study in detail, that can be done by using 3D printing with the bioprinting technology. So this is generating an example that I'm trying to show to you. So single tissue or even a larger tissues that you want to see. Say this BioX, the bioprinter have an feasibility to print your cells even in a culture plates, even in your 96 well plates, or in a how much whatever the well plates are being described in a BioX, that can be done. So you can have even a bulk tissue or a larger culture tissues, and you can use a variety of bio inks. So as if you see our previous sessions, I will be keep stressing about the bio inks. These bio inks are very important and tissue specific application. Which cell that you want to study, maybe on liver cells or heart tissues or an eye, whatever the tissue specific, that specific bio inks need to be utilized. And there are ready-made bioinks also available in selling that you can buy, which is optimized, which are having a good physiological and rheological properties, and you can utilize it. And this bioink is going to represent more about your mimic nature tissue patterns. I explored more about the bioinks in previous session. Please follow us in the biotechnica and Altem technologies to more know about why the bioinks are important to that. So using this technology, this is a one of an example where we utilize in a bioink collagen 1, and they cultured and a tumor cells and printed tumor in a medium, and they've been testing about the behavior of tumor cells and T cells, in which period of time the cells are behaving, and when we treat with the drugs, what is its behavior. All these things that we can able to do it when we generate in a disease models. So not only disease models, even you can take in healthy tissue models. And here comes in a personalized medicines also. When you want to test it within any patient, if you want to do that technology, see if a person is having a resistance to many drugs, and so we have to treat that person to save his life or his or her life. We can take a tissue from that person and you can culture it and you can print it with the bioprinters and see and screen your drugs with a maximum number of drugs and see which is not resistant to them. That will help you to understand the behavior, but also one can treat with this medicine. So here comes the role of a personalized medicine when we use this technology. Okay, how to print this? So now I think you'll be very eager to know how do we print this 96 well plate in a 3D printing model. Here's a small video for you. This is an initial bioprinter established from a cell line. Now it has been more advanced towards an BioX to BioX6 and BioMDX. This is the first bioprinter where they have started and showing from the right beginning it is feasible to print even 96 cell plates. Let's see this. Incredible. You can see that now they loaded with a 96 well plate, and then they loaded with a bio ink with the cells that will print our cells. see that initially any bioprinters that need to be have a calibration. So after doing a calibration, you can have an print your cells after loading your bio ink annotations. So you can see this print. We started printing it. So if you look into it, when we go for instance, I just say about an entity assay that you want to perform, we used to take this 96 well plate and we load it with an pipettes, and we used to have lots of instrument around us. But one such a machine is going to do all the works. That's how it's going to be automated and transitioning towards a new technology. So one by one, it will keep on trying to split the cells. Each and every cells it will try to cover. Each and every well it will be covering, and then print the cells. And moreover, if you want to print with the two different kinds of, say for instance, you want to test it for an skin cancer where we have a more layer of cells, even that is also feasibly can be done even with this, because Incredible will try to have a two different print heads, Bio X will have a three different print heads, and the Bio X6 will have a six different print heads where we can have a complex structures can be done in each different wells. That will help you to feasibly understand about a more complex structure and the behavior of the drugs. So now you can see that how minutely it is printed with an bio ink with the tissue samples, which can be utilized for screening of an drug. How many well plates that we have, that doesn't matter, you can print it. And moreover, we have different ways of printing your cells. Even this is an example. This is called droplet function printing. It will print a 3D tissue model using droplet printing. You can see that the pressure can be maintained, and if you see exploring our webinars, how much pressure, all these things can be monitored and can be controlled by introducing and using this cell in using a geometrical codes. And when we use this droplet printing, it have a more advantage, and you can print a more or different types of cells. And if you want to screen your drugs, we call it a high throughput screening technology, where we have want to have 3D tissue models and to mimic exactly like an in vivo system. This droplet printing will help you more to understand, and also will help you save your solutions as well as the concentrations of your whatever concentration that we are going to utilize it. So everything it's going to be safe and but also give you reliable results. That is the most advantage of it, and it is having a good potential for you to understand the drug behavior and screening of a drugs. Hundreds of drugs you want to screen, of course that can be done by utilizing by using a droplet printing. And moreover, if you want to understand the survival of the drugs and it be controlled by cell suspension, that is also can be done by using this one. And further, one can understand the communication and maturation of the cells. So one of the solutions when you see in Incredible Plus that is printed perfectly I gave you, and if you want to do it using in a single drop models, yes, that is also feasibly done. This is a BioX which is showing a printing. One second, I will show this. Droplet printing, how much drop you wanted, how much size you wanted, everything can be controlled under the can be controlled and you can utilize it. Okay. So now, as we go going on, is we can have a sequence. First, generate your disease models, and then print it. And once you print it, what is next? We have to go for a disease induction compound screen. So this is using a bioprinter and bioinks. We have been printed our cells. Now we have to see how it is similar to the bioactive compounds. You can see very well that this has been tested for 2 different diseases, and it is trying to show on how much treatments you can get red color or dead cells and green or live cells. As we keep increasing the concentration, there will be more dead cells that keep on emerging. And when we introduce a 3D model, that will help you to understand more about an exact node of in vivo system and accurate measurements of your concentration of the drugs that need to be treated also. So this is on a you can have an estimate of bio you can test it and accurately dose it. That is very important. Concentration of treating a disease and rapidly generate of any dilution series also you can do, and thousands of compounds. That is very important of a high throughput screening technology. Thousands of compounds you can test it and you can have a live image about the behavior changes in control and in treating with a 1 millimole or 2 millimole or 500 millimole. So like this, we have different concentration that also can be monitored. So as of now, you see only a methodology that we followed. Now you have been cracking your mind how efficiency this 2D versus 3D. Let's see a comparative results which has been published in a Cell Link, which has been stating about 3D cell culturing technology with the 3D models will have more advantages when we compare to 2D and especially in a treatment of a cancer. So let's see those because we know that cancer is a complex disease, and then treating of a cancer is a very hard task, and how this 3D technology is going to help to accelerate the drug discovery process. So this is a step, and you can see that this is a 3D model being capsulated in a tumor microenvironment. So you can see the bioprinting is going to open a new door for all the researchers, as I said, in the workflows, and also going to give in a more relevant physiological model and understand a longer culture and then of your results, and then you can come to know about its behavior also. So this is a simple step. So first you have to harvest your cells. It could be any cells you can buy it from a cell from any cell culturing laboratory, or also you can take it from a patients and embed within a bioinks. So you can use a gel or collagen. That is totally depend upon the environment of the cells that we are going to utilize it. And bioprinted within a 3D microprinters. So you can have in a bulk culture, or also you can have it in a droplet printing, whatever kind of a printing that you want. You can use that technology and you can print it. And then we are going to use an iDot. This is one new thing that we'll be introducing over here that will help you to understand the dose dependent 3D models. So what is the behavior? We are going to see automatically it is going to print and show you and stream the results of the 3D environment. Everything is going to be automated, and just we are going to monitor the results. That's it. So to accelerate your drug discovery environment, so it also need to have an side effects. So examining the side effects is very important when you go for the drug discovery process, because when you put any drugs, definitely there will be a zone of inhibition, and definitely there will be a minimum number of inhibition. But over a time of period, there will be every cells will try to attain the resistance. So that can be analyzed or can be monitored or examined only when we have a long-term studies. So when we use only these transition technology or towards in cell culturing, that will understand more about the thousands of compound effects and give you a wide range of assays and any kind of assay that can be done and efficient for the screening of your drugs. And moreover, as we stated, everything is going to be automated. You can see an automated cell living that can be done with a bioprinting with 3D cell culturing and better activity response to a 3D rather than 2D. So this is how why we need to accelerate drug discovery and development. Let's see there's how this IDOT technology is utilizing to it when you want to screen this. So first you have to own your goal and then take your 500 ml reservoir volume and then just pour it. So very minute amount of sample that we are going to utilize it, and we are going to place a plate. So it's compatible up to 1536 well plates. So drop detection that you can monitor, and you can screen droplet size as small as 8 nanoliters. So this kinds of high throughput screening will save your time, money, and also the drug concentrations that is applied for fasting your technology. So low volume dispensing is very important, and we no need to have any pipette to do this, and we can save our reagents and consumable cost, and also it will increase your reproducibility of your results and assay values. So that's why we'll be looking for a multiplex low volume dispersing high throughput screening technology, one of the products from the cell link. So these are the solutions. So with the 3D model, and they have been trying to understand the behavior of the cell lines when we're treating with the drugs. This is the most common drug that we come across for treating cancers, cisplatin. So when you have false positive results, so false positive results shows that drug is showing an efficacy in 2D but fail to save an efficacy in 3D. That means when we see our very good results towards a 2D cell culturing technology, as we keep moving on from preclinical testing to in vivo models and then phase clinical trials, the failure occurs. So here in the beginning of our drug discovery process itself, we are trying to understand the behavior so that we can cut off the what is the behavior and environment of the cells, and we can improve the behavior, and we can also increase the efficacy of the drugs. And moreover, you can see the cisplatin kills the cancer cells in 2D and fail to show the effect in killing in a 3D. And this figure conveys you the risk of false negatives in 2D. And at the same time, when you see here, so this figure will try to explore that and a false negative. As you can see, again, you can 2D cell culturing, concentration of a 3D within a collagen 1 and gelma has been lower than of the 2D cell culturing. This indicating that Jeff causes more death in 3D printed tumors and in 2D cultures. So now we have an everything is seems to, so why it is being showing good in 2D and not showing results in 3D, and 3D why not in 2D? So it's all behavior of physiological models that we're trying to give for drugs to behavior. So instead of utilizing a 3D cell culturing, then use a 3D cell culturing that will be more relevant to understand the reliability of the results. And you can see the comparison of the results and the viability of the cells, and a breast cancer as well as the pancreatic cancer cells are more sensitive towards in a Jeff treatment in a 3D than in 2D. And then results shows that, and again that breast cancer cells are more sensitive in this. You can see that sensitive in a 3D and then in 2D, and the pancreatic cell. So each and every compound, everything automation, right? So you no need to even upload a graph. So once you load your drugs and then so screening and connect with the software and going to monitor, that automatically maps and gives you the behavior of the drugs. And moreover, when you see this results, there is an opposite effect of an 2D versus 3D. One drug in a one cancer cell line, it is showing good in a 3D, but the other one in opposite way. But however, when you see on a 2D, it is showing a very good behavior in a 2D, but not same effect on the 3D. So that shows an elevated level of drug resistance in 3D models. At what time, at what concentration, that we can monitor and we can understand when we go and compare with the different cell lines or different drugs. So, moreover, you can understand more about dose dependent when you come to the accelerated drug discovery process. So, you can see that cell specific response of when you see this four graphs as you keep showing on the previous slides, you can see the dose dependent and then the cellular response of an drug cisplatin and gefactinib treatment with the 2D and 3D tumor models. And when you want to screen with a many number of drugs, you can go for a bioprinting tumoroids, where we have a 3D models of drug screening. And instantly, you can understand, one can understand about an false negative and false positive predictions, and help you can scale up for an tumor protection with an BioX for high throughput drug testing methodologies. And this is in one example. And this is on a tumor that that I monitored on a day 11. That is for the calcium PA assay for 3D cell culturing technology. And you can see they utilize an, different bioinks and then a different cell lines and a control versus an cisplatin and then other project. And you can see in the control, and when you see in a non-control, you can see that there is no, the cells are keep aliving when you keep adding your drugs. The red indicates dead cells and green indicates the live cells, and the variations in the morphology and extracellular matrix. So this one we cannot observe when we go for a 2D cell culturing. And it is more important to know about an anti-tumor effect of the drugs on different cell lines, because one drug having a good affinity to treating a one particular cell lines, but other cell line is keep on resisting, or other drugs keep on getting resistance in one different cell, other cell culture. Why it is happening? So we can make a comparative analysis of understanding the drug behavior and then pathogenesis when we have only high throughput screening technology, and also that need to be carried out parallelly on the same day. Then only we can understand the behavior of the cells. This will help you to understand even more about detailed behavior of your drugs. And this is a one which I want to show you this. This is an one which is being taken for controlling of cells. You can monitor the cells as well as an conflicts. So control, and when you treat the different drugs over the time, you can see the behavior, and also when you put the drugs in the cell lines, you can monitor this. So when we see everything on live and automated in a time over a period of time, so you can understand the behavior of the cells very easily. So that's why we will be depending on 3D models more rather than on 2D cell culturing models. I hope now you understand more about why we need to accelerate the drug discovery process in 3D cell culturing technology. And these are our selling solutions where you can print it initially with the Incredible Plus. You can see that BioX for printing with the 96 validates. Even if you want to print a complex tissues, you can go for BioX6 and then BioMDX and BioMDX Plus. And even if you want to print a more complex vascular structures within like a blood vessels along with the cells, that can be done with an DLP technology using Illuminates. And further, I think as I stated, this is a convergence technologies, right? One technology alone cannot give you the solution. So we have other synergetic solutions using a biodia. You bridge your technology in silico with in vitro from a target discovery, lead identification, optimization screening, 3D printing and going for 3D organizations, organoids or grafting. So where in a target discovery, we are going to use right from a sequence and sequence towards a structure towards a site identification, and then moving to 3D printing models. So bridging of a technology is very important by introducing or convergence of various different fields. How do we implement it? You can, when you go for a BioVR discovery studio, you can implement utilizing various kinds of technology, maybe a ligand-based drug designing, or rational-based drug designing, computation pharmacological studies, structure-based drug designing, one software as a solution for the various computation biology and chemistry. Even if you have a lots of molecules that need to be screened initially before getting into a 3D printing models, that can be screened, and even we have a specialized machine learning algorithm and artificially trained algorithms for testing your drug molecule behavior in a top-cut analysis and animal models. We have a regression based and categorization models to understand more about the top-cut behavior and pharmacokinetic behavior of the drugs. And additionally, we have a molecular simulation tools to understand about the simulation of your cells and atoms in your various environment and the drug interaction in the water environment also in using explicit solvent or implicit solvent technology. Or else, on the other hand, you want to do a structure-based drug designing where we know the active site. Maybe an instance I can say that SARS-CoV-2, where we have in a protease or playing a major role, and then we have a catalytic triad where we can concentrate, want to block that one, bring a structure-based drug designing, and introduce a screening of drugs, compounds, and get into computational pharmacological studies, and implement the technology parallelly to accelerate your drug discovery process. So, what are the other capabilities of Drug Discovery Studio? Discovery Studio has the capability of doing all this function in a single platforms, right from macromolecules to small molecules. You can do the simulations, free energy permutations, replacement, ritual screening, library enumeration using a scaffold hopping technology or reaction-based technology, QSCR, 3D QSCR technology, QCR model, EDMET screening, pharmacophore profiling, and moreover interaction pharmacophore generation. All these things have one platform solutions. And you want to do a receptor like an interaction studies, we used to know only one docking study to perform this analysis, but we having even a pharmacophore restraint based technology to understand the interactions of a pharmacophore of your drug along with your active site protein amino acid behavior also. And people who is in more interested in X-ray crystallography study, even for that also we can have a tools to understand the protein, and we can also know about the behavior of the cells. And X-ray crystallography will help you to understand the crystallographic data more detailedly. So these all are the capabilities of Drug Discovery Studio, BioCare Discovery Studio. And this is an example I want to show. Since we have been discussing about accelerating drug discovery process, usually any tool that we utilize, one drug and then one drug target and one or maybe a multiple drug that usually we will try to see what will be the effect of binding energy and so on. But in a discovery, BioVR Discovery Studio, we have a tool called Ligand Profiler that will help you to understand the multiple interaction and replaces a classical way of screening of your drugs. So multiple pharmacophores can be taken of your drug targets, and the multiple compounds can be taken. So this is a kind of a screening where you can understand the mapping of a compounds. More a number of mapping of a compounds, you can see the variations in colors. The more red color indicates the higher affinity of mapping of a compounds with a particular drug compounds. So this is also a kind of an initial level of screening before getting into 3D models. If you screen a drugs like this and we follow the steps, that will help you to save our money and also enhances your drug discovery process. That will lead to even prevent your failures in preclinical testing and the post-clinical marketing. So we have scaffold hopping, search databases, visual screening, ligand profiling, post-filtering, fragment-based drug designing. So that's what I say, that one software as a solution for multiple problems or multiple issues that we have, scientific issues. This is the one software will have all the solutions. And significance, this, that we have lacks of drugs, drug likeness, toxicity, bad process, all need to be screened, and that to be integrated in a disciplinary mode, that have an physiological experiment need to be done, and supports in the modern research and development, and a computation and cell-based assay with 3D printers, with 3D printing technology will help you to implement and enhance your research and production. And moreover, this can be used to funnel off screening of our molecules, and it will help you to enrich your results in a fast pace in a reliable manner. And you can screen a millions of compounds. Also, you can use a machine learning algorithms to understand our specificity, sensitivity of our fault proof, and prevent testing. And if you're going to implement this technology in future, in your real wise, or is going to be in a very specifically, it will replace even animal, prevents and introducing of in vivo testing in animals also, and flourishes your research culture. Ideally, one lab should have both the technology, that should have a valuable paradigm shift in your science and technology, especially in life sciences. So conclusions. If you have millions of compounds, first screen with the visual screening, do lead optimization, and then take yourself, generate your 3D models, use a high throughput screening technology, live monitor. This is a solution for drug discovery and development accelerations. So where and all you can use discovery students from a small molecules. You can have an agrochemical screening and biotherapeutics for antibody designing, enzymes for detergents, food industry for flavoring technology, biofuels for ethanol, and biodefense for anthrax and smallpox, which is already implemented, and the people are utilizing our tools to enhance the research process. So this is your question for you all. I think now you all been very eager to follow us on various social media, Facebook, YouTube, and Twitter. And here are your questions. Question number 4. The combination of technique to accelerate the drug discovery process. A. MTS and HTS. B. HTS with LTS. C. VTS with HTS. D. STS versus VTS. So scan the answer and print the answer and try to win our bio quiz, and we'll be getting excited prizes. So thank you all. So one platform for all the life science solutions from Altem Technologies, computation biology to chemistry, where bio discovery studio, 3D bioprinting with a bioprinter cell links, bio big data analysis using pipeline knowledge. So again, I show my question over here. So creatures and see the question and try to win our bio quiz. Thank you all.
All right. Thank you, Divya. This was really insightful. Hey, people, can you hear me? Please confirm in chat. Yes? Okay, wonderful. All right. So, with this, we come to an end of our 5th session, but I am sure this particular session has opened up your eyes and shown you directions in which you can pursue research, in which direction you can take up your drug screening process. And I would like to thank Divya from the bottom of my heart. Thank you so much for a wonderful session. You were very, very explanatory in your talk today, and that has helped our audience immensely. And at the same time, there were a lot of questions. I believe around 15 to 20 questions our team has shortlisted, and we have very less time. So I'll quickly take 3 questions, and after that, what we will do is we'll pass on these questions to the Altem team. Probably you can come up with the answers later on, which can be published on the website. Right. So the first question, I think this was the most popular question. That's why I'm taking up right away. And I believe it was asked by Achal first. And it was, is there any possibility of getting a hands-on or an internship at Altem or something like that, so that they could get a taste of the bioprinter?
We are trying to adopt it right now. Don't have a hands-on, but we are sure in future we are trying to. We have a bioprinters in our Altem, but we are trying to introduce it as soon as possible. We will try to reach, and we will tell about the students can also come and access, and we can utilize this technology.
Right. Okay, so there you have it, Anchal. So the next question is from Gerlina, which is, how could bioprinting be applicable in coronavirus modeling?
Very interesting. Thank you so much for asking this. See, in the cell line, if you have, for instance, we know that spike glycoprotein is a major causative agent for this to attach your human organ, right? So you can generate the disease model of coronavirus, okay, using a 3D printing. Say, take your cells and mix with the bioink, specific bioink. And recently, there are more publication in a cell-link bioprinting technology in a coronavirus itself. So see that in Altem via through cell link, where we have a published papers, will give you more insight knowledge about screening of these drugs also. So very simply, I will say that. So take whatever disease model, what we need initially the cells, and what we need to mix with the bioink, put it together, create a disease model, and then you follow that. Once you print it, screen with the drugs, and understand the behavior using IDOT technology. And to know more about the research, please look into an Altem Cell Link page where we have an newly published papers, especially in SARS-CoV-2.
Mhm. All right. Thank you so much, Divya, for that answer. I hope that answers your question. Um, the next one we have is from Ananya, and that is, does bioprinting help in producing organoids for drug screening?
Yes, of course. You can generate the organoids, and we have an special plates to generate that organoid also. And then you can also bring in tumoroids, and everything is feasibly can be done by utilizing a small instruments or small kits are there to generate the organoids. It's feasible.
All right. So these are the three questions we decided to take up today. Now we already have around a quick queue of 18 questions which we have shortlisted from the chat. So don't worry, we will take these up in a special session, or maybe we'll ask the Alton team to answer these and we'll post it as an article on Biotechnica. Right. So without taking much time of yours, guys, so this is how we end the fifth session of Altum and Cell Link Bioprinting. And today's topic was drug screening. And our speaker was Divya, and she has helped us immensely in this. Now, I have a small request. The feedback form will be given to you right now. Please fill out your feedback form. Please make sure you are filling out, because if you're not filling out, then you're not eligible for the e-certificates, which will be given at the end of sixth session. Okay, so that's very important, everyone. Now, moving ahead, we also have a couple of more webinars lined up. Announcement you will see shortly on our website. Please follow Biotechnica. If you're not subscribed, please subscribe. If your friends are not subscribed, please ask them to subscribe, because if we all are together at one place, that's when we all become stronger. Single-handedly, we are weak, just like this finger. But if we all come together, that's how we become strong. At Biotechnica, we firmly believe that if we all come together and become a fist, this bond which we build, we call it as Biotechnica. And this bond will help us not just survive, but thrive and grow bigger and better. You are the future superheroes of biosciences. You are the warriors who are going to prevent or fight out the future pandemics. But if you give up today, we are going to get defeated before we start, right? So with these thoughts, I wish to end this session. Thank you so much for attending, and it was immense pleasure hosting you all today. I'll see you soon again in our next session with Altem. Thank you so much, Divya, and thank you, audience. We see you soon. Bye-bye. Take care.